A gene mutation refers to a permanent change in the DNA sequence of a gene. This alteration can affect how the gene functions or is expressed, potentially leading to changes in the traits or functions of an organism. There are two causes of mutation. First one is environment factors or mutagens. The second one is errors during DNA processes in the genetic code, environmental factors or mutagens. One example is ultraviolet radiation from the sun. When DNA strands are hit by UV rays, they can form thymine dimers, causing disruptions in the genetic code. Next, pollutants and chemicals like industrial chemicals can also damage DNA and causes mutation. Finally, compounds like tar from tobacco are known carcinogens. These substances can induce mutations in DNA and can lead to cancer. There are three types of errors during DNA processes. First, during DNA replication, mistakes can be made by DNA polymerase as it copies the genetic code during cell division. These errors may result in mutations if not corrected by repair mechanisms. Next, during transcription, RNA polymerase converts DNA into RNA. Errors can happen if a base is skipped or added incorrectly, leading to a frameshift mutation in the RNA sequence. Lastly, for the translation error, ribosomes may misread the RNA codons, incorporating the wrong amino acid into the protein.
In this part, we are going to look at the type of gene mutation. The first type is point mutation, second chromosome mutation, and third gene mutation. What is point mutation? Point mutation is a single nucleotide base and DNA sequence is altered or changed. You guys must be wondering, how does it look like? Here is the example result from point mutation, which is sickle cell disease. It is the result from one nucleotide substitution occur in hemoglobin gene, resulting abnormal hemoglobin.
Next, for gene mutation and chromosome mutation. Many students are confused between these two. Gene mutation is a change in the sequence of nucleotides in a single gene, while chromosome mutation, on the other hand, is a large-scale change in the structure or number of entire chromosomes.
Before we dig into more detail, take a look at here of a normal structure of gene and also normal structure of chromosome for a normal people, without any mutation.
Normal gene sequence, starting with DNA, which is transcribed into mRNA. The mRNA is then read in groups of 3 bases called codon to produce amino acids. Then, here you can see an image of proper structure of a chromosome, including its centromere. So here is the listed type of example for gene and chromosome mutation that will be discussed more on the next slide.
Substitution in gene mutation refers to the replacement of one nucleotide base with another in the DNA sequence. This type of mutation alters a single codon and may affect the protein produced.
Refers to a mutation that causes severe and harmful effects on the organism. These mutations significantly disrupt the structure or function of a protein, often leading to serious diseases or even lethality. Translocation mutations can disrupt genes at the breakpoints, potentially leading to genetic disorders or cancer. Moreover, an inversion mutation is a type of gene mutation that occurs when a section of DNA in a chromosome is reversed. This changes the genetic code and can make it more difficult to read. Furthermore, an inversion is also a type of chromosomal mutation in which a segment of a chromosome is reversed end to end. This means that the genetic material within the inverted region is flipped mt/m degrees, disrupting the normal gene order. Inversions can occur as a result of breaks in the chromosome followed by the reinsertion of the broken segments in the opposite direction. Inversions can have no effect on health, especially if the inversion is balanced.
Morphoward, an addition mutation in gene mutation, also known as an insertion mutation, occurs when one or more nucleotides are added to the DNA sequence of a gene. This disrupts the normal sequence and can alter how the gene is expressed or how the resulting protein is made. Then, for duplication in chromosomal mutations refers to a type of structural alteration where a segment of a chromosome is copied and inserted into the genome, leading to multiple copies of that segment. This results in an increase in the dosage of the genes within the duplicated region, which can have various biological and phenotypic effects.
We move to deletion. A deletion is a type of mutation where a segment of DNA is lost, either within a gene or across a section of a chromosome. This loss of genetic material can disrupt normal gene function and lead to various genetic disorders or developmental abnormalities. A gene deletion occurs when one or more nucleotides are removed from the DNA sequence of a single gene. Chromosomal deletion involves loss of large chromosomal segments, affecting multiple genes and leading to broader phenotypic effects.
I would like to talk about effective mutation. There have 4 types of gene mutation: neutral mutation, beneficial mutation, harmful mutation, and lethal mutation. The first type of mutation is neutral mutation. Neutral mutation have no significant effect on the organism's phenotype or its function. For example, silent mutation, the altered DNA sequence does not change the amino acid produced. Next is beneficial mutation. It gives the advantage to organism which potentially improve the survival and reproduction. Harmful mutation. Harmful mutation negatively affects the organism, which causes diseases or disorders. For example, sickle cell anemia, antibiotic resistance in bacteria and cancer. The fourth type mutation is lethal mutation. It results in death, often before birth, or severely impairs survival. For example, trisomy 18, which also called Edwards syndrome. Moreover, there have many real-world examples in gene mutation. The first example is sickle cell anemia, which is a harmful mutation. It causes by a single base substitution of A to T in the hemoglobin gene. Then, it produces abnormal hemoglobin, HbS, which causes red blood cells to become misshapen. But, there has an interesting fact here: it acts as a beneficial side effect in certain population by providing the resistance to malaria. The second disorder, which caused by harmful mutation, is breast and ovarian cancer. The deletion of BRCA1 and BRCA2 genes, causing a frame-shift mutation. Both genes are tumor suppressor genes. They produce proteins that help to repair the damaged DNA, which maintains the genetic stability of cells. When the damaged DNA accumulates, it will increase the risk of breast and ovarian cancer. For the example of beneficial mutation, it is lactase persistence. It mutated in regulatory regions of the LCT gene. Thus, it has the ability to digest lactose in adulthood, especially for the populations with a history of dairy farming.
This image shows examples of chromosome mutations that can impact development and health. Down syndrome occurs when there is an extra copy of chromosome 21, resulting in 47 chromosomes instead of the usual 46. Klinefelter syndrome affects males who have an extra X chromosome, leading to an XXY arrangement. Turner syndrome occurs in females who have only one X chromosome, XO, instead of 2 sex chromosomes.
In conclusion, understanding these mutations is key to advance medical research and treatments. That's all from our group. Thank you very much.